right, it's my pleasure to introduce Dr. Bruce Hungate. For those of you who weren't at his talk last night, Dr. Hungate is a Regents Professor in the Department of Biosciences at Northern Arizona University, and also the Director for the Center for Ecosystem Science and Society. And we're really lucky to have him. He's done some extremely important and field-advancing work in the areas of biogeochemistry and ecosystem ecology. And today he's going to talk to us about the microbial ecology of soil carbon.
Thanks a lot, Bonnie. Sounds like the mic is working. It's okay? Okay, good. I won't have to shout too loudly then. Again, it's great to be here. It's been really fun to visit your campus and to meet many of you and talk about exciting science. Today, I'm going to talk about, as Bonnie said, the microbial ecology of soil carbon. And I like to start with an analogy from art. So this is Kandinsky's painting. It's called Fugue. Fugue is a musical form that starts with a very short subject that is then repeated and developed in different voices into, it's really the archetype of counterpoint, the pinnacle. Think of Johann Sebastian Bach. When you think of the fugue, he was really the best at this. And what I like about it as an analogy for art is that you start with something for science, you start with something very small and it scales up into something very, very large. But you can actually deconstruct and figure out almost mathematically how it's composed. It's that rigorous and that precise in its counterpoint. And you can see that here in Kandinsky's painting. I think part of what he scales is the spatial visual experience. If you drill into a part of it, you see the whole recapitulated. So you can see these repeating motifs as you go up. Now you're still probably wondering, okay, where's the science here? Well, I think in microbial ecology, we are challenged by this scaling problem. We are challenged by the notion that our reductionist science that's drilling into these tiny microbial communities is actually relevant for larger scale ecological processes. And that is an open question. It might look more like this. It might look more like Miro, another of my favorite artists. This is blue number 2, and if you try the same scaling exercise, you completely transform and lose the emergent property. So how would that work in scaling microbial ecology? It could be that carbon storage around the globe in soils is dictated more by organomineral interactions. And microbes are just noise in that much larger signal. Or it's dictated by a larger climate signal. Those are good questions. They're open questions. It's incumbent upon microbial ecologists to make the case. In order to understand the global carbon cycle, we actually need to drill in and understand soil microbial communities. And I think it's kind of hard to admit that that's true. I still think that's an open question because it potentially says, well, maybe if the answer goes one way, your science isn't all that important, at least not for the global carbon cycle. There are many other reasons we might drill into microbial communities, but this is one scaling challenge that that really integrates, unifies, and drives a lot of the work we do in my lab group. One of the challenges, I think, is a cultural scientific challenge, and that is one of embracing a quantitative approach to microbial ecology. So one of the messages I want to convey to you today is that I think we need to do a lot more. We need to do far more ambitious in interpreting molecular data with a quantitative eye. I think that those quantitative approaches are emerging, and I'm going to try to make a strong case for one. I'm not going to answer, is it Kandinsky or is it Moreau? We don't know. I'm rooting for Kandinsky. I think the world of microbial ecology ought to be scalable from the small to the large. But I'll try to give you a sense of where we are with that. So I'm going to start with this experiment in Florida. I spoke about this a little bit yesterday about soil carbon and net primary productivity. Today we're going to really use that as a starting point and dive into the soil microbial community. But for those of you who weren't there yesterday, this is the scrub oak ecosystem in central coastal Florida. The site is located at the Kennedy Space Center. And from 1996 to 2007, we built a CO2 experiment on this site. So we had high and low CO2 for about 12 years. You can see the plants here are oaks. Maybe you can't see that, but you'll believe me when I say it. This is a saw palmetto. And the system really has an impressive soil horizon. Most of the biomass is below ground. It's a fire-dominated system. So we started our experiment after a controlled burn, and then we built open-top chambers on top of the regrowing vegetation with or without elevated CO2. So these fans blow either normal air into these chambers, or maybe the one over here blows normal air with an extra bleed of CO2, simulating an atmosphere of about 2050 to 2080, depending on how much we burn fossil fuels between now and then. One of the questions we asked in this experiment is, will elevated CO2 increase total carbon storage by this ecosystem? And this is a common question in CO2 experiments because the expectation is when you give plants more CO2, they'll photosynthesize faster, they'll grow faster, and as they die and decompose, you'll actually accumulate more soil carbon, together increasing the carbon content of the system. Here's the results at the end of the experiment. Those stacked bars show the total carbon content in the ambient and elevated CO2 treatments, ambient on the left, elevated on the right. And you can see the colored bars are the colored parts of the bar are the plant, and the gray is the soil. So if you focus just on the plant, there's quite a CO2 effect. It's pretty large. The plants did grow more in this experiment. It was statistically significant and all that. But when you add up all the carbon, including the soil, it's not significant. And that's what's shown here. Really, the two bars aren't much different. There's a little blip higher with high CO2, but it's obviously not statistically significant. So is that the end of our story? CO2 didn't increase sea storage, but it turns out it's a lot more interesting when we look at some of the details of the soil response. So here's one look at that to begin with. This is a picture of the light fraction of soil carbon in elevated CO2 in our experiment. So the light fraction literally is light. It's low density. It's the part of soil organic matter that floats to the top of a solution that we concoct that has a density of about 1.6. Why is that important? Well, that light organic matter actually cycles fairly quickly. And it's the organic matter, the part of soil we expect to be most responsive to something over like a 10-year time scale. Because a lot of soil carbon, of course, is much, much older than that, thousands of years old potentially. So we focused on the light fraction. And when we did that, what you can see is the elevated CO2 here in the squares, ambient in the circles. This is the time, the beginning to near the end of the experiment. Actually, that is the end. And light fraction soil carbon as a pool size. They start about the same, and you can see over time the high CO2 is actually lower than the low CO2. Exactly the opposite of what you would expect when plants are growing more and putting more carbon into soil. So why does that happen? The best explanation for this is something called the priming effect, which I'll explain in a minute, and that's kind of the point of departure for diving into the soil microbial community. I won't do that prelude yet. Okay, so the priming effect, what is it? First, we start with soil respiration. Soil respiration is the process by which microbes convert organic matter to CO2. And it's shown in this cartoon, microbes chewing on soil organic matter. Of course, they're building biomass, making ATP, but in the process, they're releasing CO2. If we add a labile carbon substrate to that soil system, the microbes will eat that too, and they'll produce more CO2. The interesting thing, and what we call the priming effect, is that when you add that labile carbon, you actually stimulate the decomposition of that older soil organic matter. This has been observed since about 1920. That's the first report of this phenomenon that I've found. And again throughout the literature, you see it. It's had a sort of resurgence in the last decade or so of attention because people are recognizing, you know what, maybe this is happening out in natural ecosystems, not just in the laboratory as some sort of artifact. And indeed, there's plenty of evidence that that's the case. This has led microbial ecologists to ask why, what's going on with these microorganisms that would cause this priming effect. And I will note that right away, or right here with the priming effect, we've already violated how most soil organic matter carbon cycle models work. They don't allow this sort of interactions of different pools. So this is interesting to think about and to wonder how it's important. It's possible to measure the priming effect, and we do this using isotope tracers. What's done is you add a carbon-13 labeled substrate, and then you monitor the production of natural abundance CO2, here abbreviated as 12C. So you add your tracer and you watch what happens to your unlabeled soil pool, and you can partition out exactly how much priming you're getting in your experiment. This is how we measure it. So, as I said, microbial ecologists have come up with lots of hypotheses. Why is this happening? One of them, probably the most prevalent, is one I'll call the two groups hypothesis. And that says that there's a particular group of microorganisms that's responsible for responding initially to that labile carbon substrate that we add. And then there's another group of microorganisms that through some chain reaction or signaling or resource shift is decomposing more soil organic matter. You see this again and again. Sometimes it's cast as the copiotroph oligotroph or the RK, all kinds of names given to these different groups, completely, I might add, in the absence of evidence. So this is a wonderful hand-waving field, this whole priming effect. I'll be guilty of some of it today, but I'll try to at least identify it. So how might we figure this out? How might we test whether these two different groups are actually involved in priming? And the answer that I'll offer today is stable isotope probing. Stable isotope probing works like this. You add an isotopically labeled substrate, and the organism that consumes that substrate incorporates that heavy isotope into its DNA. Then you extract your whole community DNA, and through a step I'll show you in a minute, you can identify who's taken it up and who didn't. And it turns out stable isotope probing has been used really powerfully for lots of cool questions, including things like who's using glucose, but also who's using methane and carbon dioxide, all kinds of xenobiotics in the environment, all kinds of specific substrates. That's been the primary focus of this stable isotope probing approach. Who's using this substrate, or the other substrate? And that's great for our priming effect question for the first part, right? We can figure out who's using our labile substrate. But for the second part, it's not quite what we need. We need something else because labeling soil organic matter is really hard. And really what we want to know is who's changing their growth rate. At least this is a start. So for that, we use labeled water. Because organisms that are synthesizing new DNA incorporate oxygen from water into their DNA. And so if we add an isotopically labeled form of water, like 18O labeled water, we can identify who's growing. So that's a new twist on stable isotope probing that's been really powerful for us. So how do you identify who's using, who got labeled in these stable isotope probing experiments? You don't use a mass spec. I'm a big fan of mass specs. They're great for measuring isotopes really precisely, but for this technique, we go very old school and actually use a centrifuge. So this is a cartoon of a tube of cesium chloride, which has the property that when you spin it in a centrifuge, creates a nice density gradient that spans the range of densities of nucleic acids, like DNA. So what you do is you put your environmental sample in the top of the tube, spin this in an ultracentrifuge, and the heavy DNA will actually migrate down the tube towards the denser region, whereas the light DNA stays up at the top. This cartoon shows what you'd get from a pure culture experiment where some of your cells didn't grow and some of them did. It's a really nice, clean separation. What you usually find in a complex natural community is more of a smear like that. And that's because organisms vary in GC content, right? Guanine cytosine content, and that has a huge effect on the density of DNA. So we need to account for this kind of background variation in DNA density. And that's done as follows. You might make a graph like this with your control tube that got no isotope label. When you add your C13 label, you see a slight shift to the right, like this you can see in the pink trace. And then you'll say, and this is what's done, okay, this is the region of DNA that took up the isotope tracer, and this is the region that did not, heavy and light. And you see this all over the stable isotope probing literature. The point I want to make here, this is powerful. I don't have a qualm with it, except that it's taking a qualitative approach to a potentially quantitative solution. So keep that as a placeholder for a minute. But we've done this, and we've combined a carbon tracer like this with our 18O water tracer that I just showed you in experiments where we get results that look like this. So you can imagine now an organism that uses glucose for growth. We'll call that the star. And when we look at where it sorts out in our density gradient without any added isotope, it's right about there. Then when we add glucose, we see that it shifts. It's also growing. It's using glucose for growth. So it's growing. So we expect a signal in oxygen, right, in labeled water, because it's using glucose to grow. And we expect it to shift even farther when we give it both heavy glucose and heavy water. What about another organism that's growing in our community, but it's not using glucose? We expect it to not change at all when we add glucose to that sample, but we do expect it to shift when we add 18O labeled water. So this is the kernel that we can use in our priming experiments, because we can ask who's using glucose and growing, and then who's growing but not using glucose. And especially when we add glucose, who's changing their growth rate without utilizing glucose? That's the basic idea where we're using the stable isotope probing effect to get at this question about priming. So here's some real data from my master's student, Becky Mao, who did an experiment with C13 labeled glucose and 18O labeled water. And we used sort of the old school approach for saying, okay, there's the heaviest region, there's the middle region, and the rest is light. These guys here appear to be utilizing glucose and growing. There are definitely some here that are growing, but not obviously utilizing glucose. You know, again, we're using a qualitative approach, and it's definitely subjective. What she did in this experiment was a priming experiment where you add repeated pulses of glucose and monitor what happens in terms of oxidation of older soil organic matter. Early on, what she found is when you add one pulse, there's no priming at all, and actually the opposite happens. The organisms shift towards using that sugar, and the oxidation of older soil organic matter goes down. It's sort of negative priming. This is something some people find sometimes. But over time, we saw the priming effect after 6 weeks, after multiple pulses. And you'll see why I'm bringing that up when I show you the next slide. Because the community responses to these dynamics were really, really different. If you focus on the left here, we had the single pulse scenario. So this is when priming went down, when the microbial community really shifted towards utilizing glucose. And what we found there in the glucose-utilizing community, so these are the organisms that had both the C13 and the O18, is that community was really dominated by Arthrobacter. You can see in the pie chart here, it's over 3/4, and Victor, my artist friend, celebrated the victory of Arthrobacter by drawing that giant pile. John, this is what I was telling you the other night. Right. It was also true in the soil organic carbon oxidizing community that we had a dominant organism. It was a bacillus species in that case. Because of those explosions of these individual taxa, biodiversity went down. And you can see it's an evenness response. There wasn't much of a richness response, but evenness went way down because this one organism, Arthrobacter for the glucose utilizers and Bacillus for the soil organic carbon utilizers, really took off. We see this in ecological experiments sometimes. When you add a resource, one organism's able to capitalize and it outcompetes the others. That's probably what's happening here. But after multiple pulses and over time, we saw a really different response, a very even community. And this was actually equally even to the soil community at the very beginning of the incubation. So the biodiversity had recovered in concert with the priming effect becoming evident over time. So what can we say about that? we can say that we've seen an association of a change in biodiversity with the priming effect. This is where I'm doing hand waving, because do we know that it's causal? No, of course not. It's just that we saw these at the same time. And the field is full of this kind of explanation where, well, we saw these things happen together, therefore it must be causal. I think we can dig deeper, and I want to show you one way how using stable isotope probing. So this is a continuous gradient in density, right? It's a quantitative variable, this x-axis here. And we can look back at our star organism and imagine our blob organism too and say, well, it looks like the blob grew more than the star because it shifted its density more. But we've got this background variation of GC content that we need to account for. If we can do that, if we can make those shift the control so we're comparing them all in the same basis, then maybe we can make it quantitative. And what's exciting for me as a biogeochemist is that we can change density to something far more useful in terms of monitoring the carbon cycle, and that's the actual enrichment in isotope composition of our target organism. So we think we've come up with a way to do that. And the way it works is that if we go back to Becky's data, instead of identifying the heaviest region and the kind of heavy region, we're going to just take all our fractions and sequence every single one of them. When we first had this discussion, and Becky was the one who suggested it, I said, you are crazy. I mean, because that amplifies your sequencing costs by a factor of 20 or so. You know, in order to get what? But we talked about it more and we started realizing, well, wait a minute, we could actually do something kind of cool with this. So here's an example. Sorry, I need to go back and explain. So we sequence the 16S ribosomal RNA fraction, gene in every single fraction. And with that, we get a marker of the relative abundance of all the organisms in our microbial community as a function of density. And then what we do is quantitative PCR to quantify the total gene copies in each fraction. I'll show you a little bit the math that goes into this, but the basic idea is that if then you can calculate what's the average density across all the fractions of every single taxon in our community. And the next graph shows you an example of that with that same genus Arthrobacter. Here's Arthrobacter in a control incubation with no added isotope, and that's its density on average. Here's the weighted average density. What happens when we add 18O water? It shifts to the right because it's growing. And then what we can do is actually measure that shift and run it through our mathematical model to turn it into atom percent C13. I don't want to go through the details of all the equations, because that's torture in an environment like this, right? But my point in putting them up here is twofold. One, the math is really simple. It's basic algebra, calculating weighted averages and things like that. Number two, this is borderline felony conduct in the world of microbial ecology. I can attest, based on the content of reviews I've received from some of the manuscripts. So, and I think it really is a cultural difference where coming to this field really as an ecosystem scientist, said, well, we can assume this and we can assume this and we can test our assumptions and we're probably wrong, but maybe we're wrong in a useful way, or maybe we're not that wrong, or maybe when we propagate our error responsibly, we'll be wrong in the way all ecology is wrong, right? It's an estimate. And that's what I think we need to do. So here's a diagram of the whole technique where you start with an environmental sample. You're adding isotopes, letting them incubate. It is in the lab right now. We have done some incubations in jars in the field. I'd really like to try it just in an unaltered soil. It gets expensive, but someday we'll do that. Anyway, let it incubate for a time, extract the nucleic acids, spin in your ultracentrifuge, and that, of course, separates the DNA molecules by density. And then the real difference from standard SIP is this next one, where we're sequencing every single fraction. Sequencing, qPCRing, running through QIIME, the bioinformatics database, and you end up with this whole catalog of atom percent XSC13 or 18O for every single taxon in your community. So I spent much of my PhD days collecting plant species from different experiments that had added isotope tracers, and you collect each species very carefully, you grind each one up individually, and then analyze them on the mass spec. This, to me, is really cool because it is pretty laborious, but after this process for one sample, you get 1,000 different species and their isotope compositions. It's really cool. There's another approach that's somewhat similar that I'll mention. We're collaborating with some folks at Lawrence Livermore National Lab. And they use a device called NanoSIMS, Nanoscale Secondary Ion Mass Spec. Some of you may have heard of this technique. It's called ChipSip, but I'll describe it briefly because it's also really cool and complementary to what we're doing. So in this case, you add isotopes to your environmental sample. This translucent blob is our artist's rendition of an environmental sample, be that soil or stream water or whatever. Then you extract DNA from that sample, just as before. And then you've prepared this microarray. This microarray has RNA probes on it, each one a specific sequence for a specific organism that you think might be important in your community. And then what you do is add your environmental DNA to that microchip, and the DNA sorts out and binds with the probe where it matches. So basically the way I think of it is it spatially organizes all the environmental DNA in your sample into different taxonomic units. And this is relevant for NanoSIMS because NanoSIMS, then you can look at this under fluorescence and quantify it. And then the NanoSIMS shoots a cesium beam at each little dot and monitors the secondary ions that are produced as that DNA or RNA is decomposed, and those secondary ions contain isotopic information. So again, you can get a taxon-specific isotope measurement for these different members of your microbial community. What do the data look like? They look like this. Here is QSIP, so this is our quantitative stable isotope probing. This is not the NanoSIMS approach, but the idea is really similar. Each one of these bars is an individual taxon, an individual genus. If we go to the OTU level, or what microbial ecologists use for bacterial species, basically, you get even more. The left panel shows the control case without added carbon, and the right panel is what happens when we add carbon. So when you add carbon to soil, lots of things grow, right? Big surprise. Actually, it's kind of interesting how many things grow, how evenly distributed that signal is across bacterial taxa, because most of them are shifted to the right. One of the things we've done with these data is to look to see, is there any organization to this growth response, to carbon or to the growth rates that we're observing in soil? And that has to do with searching for a phylogenetic signal. This is work that Ember Morrissey led, and I think it's a paper that some of you read. It's one I sent up here in preparation for this visit. And what we found is that there is a phylogenetic signal. So a particular growth rate exhibited by one taxon is more likely to be similar to another's if they're closely related. And this has some sense if you think of growth rate as being a trait that's selected for and propagated through evolution. So that was cool too. So I mentioned before in Becky's experiment that there were multiple pulses. Remember in week 1, we didn't see any priming. Actually, she saw pretty strongly negative priming. And then over time, over weeks, as the experiment progressed with additional pulses, we saw more priming. Ember saw exactly the same thing in her experiment. The week 1 response wasn't quite as strongly negative, but we had lots of priming by week 6. So one of the things Ember did is to combine the carbon-13 and the 18O signals in our quantitative stable isotope probing to try to figure out, to test this hypothesis that there are two groups of microorganisms, one that's responding to the glucose and utilizing it, and another separate group that's degrading the soil organic matter. And the way she did that is like this. This is complicated graph, so let's take a minute to orient to it. The y-axis is the simpler of the two axes. This is just excess atom fraction C13, or you can think of it as the amount of C13 that an organism took up. The x-axis is the change in excess atom fraction 18O. So this takes the control condition without any glucose added and determines a baseline growth rate for every organism. Then it takes the glucose added treatment and measures a new growth rate, and this x-axis is the difference. So it's the change in growth in response to glucose. And if an organism, for example, is growing and it's utilizing glucose entirely to support that growth, we expect a 2-to-1 relationship between the C13 and O18. And that's because the organisms are getting about half of the oxygen in their DNA from water. The other half actually comes from organic matter. That slope of 2-to-1 is about what we saw in week 1. There's a lot of scatter around it, and the error in the QSIP technique is pretty large, so some of that scatter is just error. I'm not showing error bars here, but the slope is right around 2. That makes sense when you think, well, in week 1 there was no priming. We added glucose, and we saw no change in the utilization of older soil organic matter. So anybody in the community who's growing more is probably using glucose to grow, and that's what that slope tells us. But when we turn to week 6, it's really different. We saw an increase in growth rate that's larger than we can explain by utilizing just glucose. That is also consistent with the whole jar CO2 mass balance because we saw priming at week 6, remember? The priming effect was really high in that 6th week. That means some organisms are utilizing older soil organic matter in order to grow. So that's priming, this is not priming. And then when we think about these data in terms of the hypothesis, there's absolutely no organization to the community in terms of who is shifting more down on that slope or who tends to stay up high. There's no phylogenetic signal. There's no, we've looked at phylum all the way down to OTU to try to figure out, is there any evidence for this idea that some organisms are specializing in that initial pulse of glucose and others are specializing in utilizing soil organic matter? And we think these data pretty strongly reject that hypothesis. So this is one example of how I think a technique like quantitative stable isotope probing can provide insights into questions in microbial ecology. So another way we're pushing this, so far I've just shown you excess atom fraction C13 or O18, and that's how far our model went with these papers. But we think we can take it farther to actually estimate vital rates of microbial species in complex communities. Again, I'm not going to go through the math, but the bottom line here is that if O18 is a measure of DNA synthesis, we can use that as a quantitative measure of growth. It's a lot like we use C14 leucine or tritiated thymidine for measuring productivity in microbial communities, another tracer, but the same exact concept. And here, the bottom line is we can measure death and birth and net growth rate. These are cool, I think, because it takes me right back to introductory ecology, right, where we're modeling populations as logistic growth or exponential growth, and we're modeling competition through Lotka-Volterra interactions. It's a foundation for asking far more quantitative questions about species interactions in microbial communities. And I feel like we're right at the surface. Here's one way or one example of a community where we've partitioned out these different growth rates. So this is gross growth rate or birth rate. The black is death, nice morbid color choice for death, right? And the green is the net, the balance between them. And this is a community right after a simulated rain event. There have been some other studies that have shown massive mortality of microorganisms when you wet up a dry soil. And actually, if you try to calculate how many of these poor creatures are dying during these wet up events, they're by far the biggest examples of massive mortality I've ever seen. These are tiny, tiny creatures, and rainfall covers a large area. Anyway, we saw that phenomenon could be captured by these estimates of vital rates using quantitative stable isotope probing. So that was cool. Another thing I've tried is, well, how might we relate this to the relative abundances of these organisms in communities? Because we'd expect those that are growing fast and dying slowly would eventually dominate the community. So maybe we're not actually getting any new information. But when we do that, here's relative abundance. So this is the typical data you get from a 16S community screening, where you're just looking at who's there. You get a relative abundance profile. And this is our measure of growth. In this case, it's excess atom fraction 018. At first, I looked at this and thought, there's no relationship. That's too bad. But then I thought, wait a minute. That means what we're getting is completely independent information from what we typically get from sequencing. And maybe we can look at this another way and try to think of what we might learn from this type of graph. So if you imagine this is giving us relative abundance, got abundance. This is giving us biogeochemical activity or importance, at least in terms of assimilating elements from the environment, because that's what organisms do when they grow. So we might imagine we've got some organisms down here that are not assimilating at all, some organisms that are growing at a moderate rate. These might be our dominant or even foundation taxa, and this might be our keystones, right? Organisms that are really important but have a low biomass, so their importance is belied by their relative abundance in the community. We might use this as a way to postulate the functional importance of different microorganisms in our community. And this is one way we're looking at this. The particular example I'm showing here are from Fremont cottonwood and Gamble oak leaves decomposing in streams. We're trying to understand how different characteristics of the microbial community on these decomposing leaves facilitate or do not facilitate transfer of energy up the food web. And finding some really interesting patterns, like the slowly decomposing leaves actually transfer more energy to insects. And we think the microbes might be involved in a key way. In another fun project, we looked at canopy soils in redwood trees. I have somebody here for scale. There he is. There's George Koch, partway up the redwood tree. These are huge trees, of course, and they're old, really, really old. What that means is when their leaves fall from high up in the canopy, some of them get captured on large, large branches. And over time, they form these canopy soils that can be a meter deep, that can support a 40-foot epiphytic tree. That's pretty wild to see a 40-foot tall tree growing in the canopy. Here's a picture of one of these canopy soils. Here's another one. That white spot is a helmet of one of my colleagues. And in one of these, at about 75 meters off the ground, this is my favorite epiphyte experience, because this is a vaccinium bush that was absolutely loaded with delicious huckleberries, so we had a snack. We sampled these canopy soils at different heights above the ground. And the idea was to envision these as canopy islands at various different distances from the mainland in order to see if we got diversity patterns consistent with the theory of island biogeography. So far, it looks like there's a tendency for richness to decline, but we're with height, which would be consistent with island biogeography, but we're still very early in sequencing and analyzing these data. I'm really excited about this technique and this approach to microbial ecology because I think it is opening up what is the hackneyed black box. You hear that phrase a lot in microbial ecology. It's just a big black box, and we don't know what's happening. Now we have the tools to look inside quantitatively and figure out what's going on. I do think one of the challenges in the field is to embrace a quantitative approach, to embrace the approach of most ecologists and ecosystem scientists in order to forge ahead without fear. Victor, when I told him about this, he gave me 7 graphs. I said, wait, that's not even close. There are thousands of species here in these communities, and that's what's really exciting about this approach. So I animated his drawing with PowerPoint. I want to shift gears a little bit now because there's another approach in ecology that I think has a nice analog in art. This is Keith Haring's painting, and here it is as re-envisioned by Ursus Verley. He has a project that I love. It's called Cleaning Up Art. And so he takes this nice complex image where you can actually see a face and, maybe there's, I'm not sure what the meaning is. To me, it looks a bit like a bull. But he says, nope, we have to organize it. We have to put all the pieces into different piles that correspond to the same colors. So what you learn from this is that this is what makes up the art. When I look at the original, I wouldn't be able to tell you there are 10 different colors. But when you organize it and reframe it, you see that. So you see some aspect of the composition. It's definitely a visual analog for reductionism because, of course, you lose a ton of meaning when you do that. And in ecosystem science and ecology, we do this. We also take another approach, which is maybe more common in global ecology and ecosystem science, where we say, that's way too much detail. We've got to squint and blur and just try to extract the main message. Sometimes that works. You can maybe sort of see a face there. And sometimes it doesn't. I think of atmospheric inversion and a lot of the large-scale processes where we're looking for something like that whole atmospheric pool to integrate information about the terrestrial biosphere and the ocean and things like that. So I mention this because I think I'm challenged in my own microbial ecology and ecosystem science work to embrace both. And here's an example I'm going to show you where I think it really does both in a way that's interesting. This is work that's led by Paul Dijkstra. It starts with this, the Krebs cycle. Don't worry, we won't go into detail. This is glycolysis, and this is the pentose phosphate pathway. This is the Krebs cycle, glycolysis, and the pentose phosphate pathway according to biogeochemists, or to advanced biogeochemists. So, which brings up the question, why do we know from Bio 101 that there's this tremendous complexity of biochemistry that is in our ecosystems, and we stuff it all. Now I'm using that hackneyed black box with a little bit sticking out, just to hint that maybe there's something there. But the first question you might be wondering is, well, let's put aside what it might teach us. How on earth would you do that? How would you measure all these different biochemical reactions in a complex microbial community? And the answer is, again, a quantitative approach that relies on isotopomers and our knowledge of these biochemical pathways. So here's pyruvate, and it's got 3 carbon atoms. If we know the position-specific location of our isotope tracer, we can consider, this would be 1 pyruvate, in the context of glycolysis in the Krebs cycle, as shown here. And we know exactly where that position 1 carbon is released in terms of that biochemical pathway. We also know this for positions 2 and positions 3. 2 and 3 actually sort of bounce back between those two so that it looks like that. And we also know how this works with glucose. If we start with a glucose labeled at position 1, 2, 3, 4, 5, or 6, we know where those atoms are susceptible to release of CO2. So basically what Paul does is he adds in series of incubations, there are usually 9 or 10 for each sample, all these different isotopomers, not in order to add carbon. You can do it at very tiny addition amounts. So you're not changing the energy balance of the organism, but you're tracing its biochemistry by watching where those carbon atoms are released. And he can put this into a model that actually gives you relative fluxes through all of the individual steps of those 3 pathways, which is fascinating and completely overwhelming. But one of the very cool outcomes of that approach is it gives you the balance of energy generation and biosynthesis. So how much is that organism using carbon in the environment to make ATP, produce CO2, versus how much is it using it to build new biomass and grow? It turns out that that ratio, called carbon use efficiency, is really important in global biogeochemical models. You can actually change the entire carbon stock of the world's soils by altering this carbon use efficiency in the models. So it's an example, if you go back to the very beginning of the talk, of the potential power of this scaling from microbial physiology all the way up to the global carbon cycle. One of the things we found with this, probably the main result, is that carbon use efficiency measured this way is really high. It varies a little bit. You can see on the right, the yellow, the peat mix, and the cottonwood litter are a little bit lower at maybe 0.57 or so, 0.55, compared to the agricultural soils from the UK. which are up at around 0.73 or so, which might not sound that interesting. By itself, it's not. But let's put it into context here with one of the recent reviews about carbon use efficiency, which says there's a maximum carbon use efficiency of 0.6, and natural systems should approach 0.3. And this is offered as thermodynamic calculations, as though it's theoretically prescribed. So we've made a lot of people angry by saying, no, actually, carbon use efficiency is much, much higher than that, and it doesn't vary that much. So we've tried increasing temperature, which I'll show you in a minute, to tweak carbon use efficiency. We've tried different resource balance, and it's very inflexible measured this way. We think we're starting to understand why people using other techniques have measured such different ranges of carbon use efficiency. But anyway, in the context, there you can see the theoretical maximum and the average. So as I mentioned, one of the experiments we did was looking at temperature response. We incubated soils at 5, 10, 15, and 20 degrees and measured carbon use efficiency using this technique. And that's shown here on the left. So an organic soil and a mineral soil, heat up the soil, what happens? A lot of models, a lot of speculation says carbon use efficiency should go down. And this is often how it's treated in the large-scale models. We found no evidence for that whatsoever. But what we did find is that microbial turnover went up. So turnover, think of anything that's causing an organism to die, like predation or stress or anything like that. How I read this is the instantaneous biochemical partitioning of energy towards energy generation and biosynthesis is pretty inflexible. If an organism is going to utilize organic carbon, it's going to utilize it in a pretty strict stoichiometry in order to build new biomass. But as temperature goes up, you might get more stress, you might get trophic interactions that cause more organisms to die. And actually we're looking now to see if we can explain these results based on predation. You can see turnover definitely goes up with temperature. Carbon use efficiency goes down. So this is one example of where this kind of microbial diving into the small details of biosynthesis, actually, and biochemistry might inform some inferences we'd like to make about larger scale processes. Another one where we're using or seeing the priming effect helped us understand why we might see this phenomenon. I spoke about this yesterday, where we see increased growth in high CO2, but it looks like microbial decomposition increases as well. Well, priming is one mechanism that could explain that response, leading to no change in soil carbon in the long term. One of the ways I like to think about this is retracing the history of global scale carbon models, where the first ones in the 70s were called the green slime models, as though one alga was the entire Earth, and that's basically how we modeled it. And then people were excited when we got to the big leaf model in the 80s, where we had stomata and gas exchange and water and energy balance. And now, of course, we have dynamic global vegetation models that incorporate plant biodiversity as plant functional types. And we maybe have a dozen or so, plus or minus, that incorporate what we think are the functionally significant parts of plant biodiversity in terms of the global carbon cycle. My dream, and many people's, this isn't unique, is to bring something like that to the microbial world. The jury is still out whether it's needed, but a lot of us are working to test the basic idea. So I want to end by commenting briefly on this bug I have about microbial ecology. Much of the field, I think, ends here, where you've done your careful experiment, you've extracted your DNA, you've characterized your community, and then you put it in an ordination plot and you say, well, before my communities were different, after, not so much. And that's pretty much where it ends. There are many, many papers I've read, some of them really interesting, that end with this. And I think that taking a more quantitative approach will improve this approach. I bet you're wondering what this is about. It's actually a cool study. So these are different roller derby teams that have different skin microbiota before the match. After the match, they've brushed skin and they've exchanged microbes, and Their microbes are different. I thought that was pretty funny. Some of the areas I've been getting into in taking a more quantitative approach involve human and medical microbiology. One example is the communities that inhabit our noses. And who among us are carriers of Staph aureus is an important question, because Staph aureus can be transmitted from one person to another, and it can be an opportunistic pathogen in the sense that it's absolutely asymptomatic most of the time, but under the right conditions can really cause a problem. We found in this work that if we used relative abundance and typical ordination, we got no clear patterns, no insight. But when we took a proxy of absolute abundance as total gene copy number by the relative abundance of each organism, we got far stronger insights. And we're looking at putative competitors with Staph aureus that might be developed as probiotics in some model mucosal systems. Another example of ecology I like to think of, I learned the definition of disturbance as the catastrophic removal of biomass, and that's a pretty good description of circumcision. And it turns out that circumcision has a huge effect on the genital microbial community, but that effect is best understood if you look at absolute abundance, not relative abundance. It turns out circumcision reduces the total community size in the genital microbiota. And this isn't just basic science. It turns out, maybe some of you have heard this, circumcision actually reduces HIV transmission, but the mechanism is not well understood. We've been looking at the genital microbiome as a possible explanation. And what this graph shows are 10 different anaerobes that are abundant in uncircumcised men and their odds ratio of HIV infection. This is actually a randomized controlled study of circumcision. Values to the right are the odds of HIV transmission with a 10-fold increase in anaerobe abundance. So it's not causal, but it's a pretty strong implication. And actually, there's even a mechanism for this, and that with these anaerobes, as their abundance increases, you get greater concentrations of cytokines, which are an inflammatory response of the immune system that we know makes people more susceptible to HIV transmission. So this is something we'll see if they bite. But it's an example of ecology, right? This is microbial ecology being super relevant to things like human health. And there are many, many others, as you've probably heard. So as I said, I don't know if it's Moreau or Kandinsky, but I'm rooting for the Russian over the Catalan. And as I look, though, maybe with a more objective eye, the best artistic analogy is like Dali, where I don't know if you watch videos of Dali talking about scaling in his art, he's so convinced the meaning is there, but most of us look and, wow, that's trippy. I don't really get it. And that's microbial ecology for me right now. Thank you. Questions? Yeah.
So you sampled canopy soils at different distances off the ground. And that was it was decreasing in species richness. What were you? Was that with microbes? The soil? Where were you? What canopy levels? How were you getting the micro? How were you getting soil from different canopy levels?
We climbed up the trees and sampled the canopy soils. Yeah. Yeah, so these are canopy soils. I mean, they're really patches of soils that are, the lowest were maybe 20 meters off the ground, and the highest were about 80 meters off the ground. Yeah, and we had a gradient of 6 or 7. We also measured the size of each canopy soil patch, because island biogeography is also about the size of the island and its distance from the mainland. It really is a long shot, because I think the standard expectation of a microbial ecologist is that that's not far enough for dispersal to be a limitation. But okay, we wanted to climb the trees. No, I thought it was a good idea to try to illustrate. Even if we get a negative answer, I think it'll still be interesting to find out. Yeah.
So if I'm understanding correctly, in your priming experiments, you found that the type of substrate the microbes were utilizing changed through time, but the community wasn't shifting in any particular way. So that implies that the microbes are deciding to eat a different compound even when you continue to add glucose. So if it's not a community shift, what do you think is mediating that metabolic shift?
Well, it's a good question. I think from a microbe's perspective, it's carbon. And, it's carbon of varying oxidation states and, energy yield. But for many of them, the dogma is that they're energy limited. So they will use what's available. I mean, that's how I'm interpreting this result. As soil organic matter, carbon is becoming more available through priming, through a mechanism that these results don't speak to. We're not seeing any organization of the types of organisms that are preferentially using that soil organic matter. So, they probably don't care if priming is happening or that we're adding this glucose, except to the extent, there's something more to eat. If that triggers a chain reaction and causes new substrates to be available, they're going to eat them too. I mean, I'm speaking simplistically because, of course, there are fine degrees of specialization within the microbial community, but we're not seeing it sort out in this construct that we're calling priming. So I think we ought to, we don't know really what's liberating the soil organic matter. I think it might have to do with organisms growing into new environments and just encountering it. Because, the whole idea of soil organic matter recalcitrance is kind of being challenged now, maybe being replaced with a spatial protection and spatial segregation of organic matter. And that's a pretty simple explanation, but it's my favorite right now. John probably disagrees.
Well, it seems to me that the obvious answer, and obviously it's not obvious to you if you look at it closely, is that you've eliminated the carbon limitation and created, say, a nitrogen limitation, and possibly the microbes are secreting more phenol-oxidases to degrade the organic matter to release more nitrogen to relieve that deficiency. What do you?
I think that's entirely possible. We don't have direct evidence to speak to it. I have thought it would be fun to go back to the density gradients and sequence for specific target genes to see if we have evidence for some of these genes increasing in response to glucose addition, but maybe using glucose to make them or using something else. I'm really looking for smoking gun type evidence that's really strong. I think it's a sensible mechanistic hypothesis. I'd like to know how to test it rigorously, I guess. Andrew, yeah.
So if everybody has similar carbon use efficiency and you add a lot of sugars, readily available carbon, and it doesn't shift the community composition, it seems like there's no niche partitioning, but there's tremendous diversity.
We do see that adding the carbon shifts the community. It changes the composition of the community. What I showed with the priming example was about there's no community organization to the shift in biogeochemistry. We're not seeing clear partitioning of two groups. So I'm not quite sure how to address your question. I don't think we have direct evidence for niche partitioning.
It seems like there's not strong evidence for niche partitioning in consumption, so it seems like the natural alternative is that there's strong top-down control.
That's a good point. Yeah, right? That would be really interesting because it's so diverse. It's huge. And I think part of the answer is there has to be some niche partitioning based on what we know about the physiology of these organisms, but maybe adding glucose is not the best way to test for that because it's not a particularly specialized substrate. Though I have heard some objections to that statement, that actually there is quite a bit of specialization and some organisms really don't like to use glucose. I think one of the things we need to keep in mind with our experiments is that the incubation period is long, on the order of days. And so you can get metabolic crosstalk as organisms that initially took up the glucose die and are consumed by other organisms that will look like they used the glucose. So I look at it as it's definitely a community impact of this resource addition and a realistic simulation of how it should propagate through the community, but we have to be a little bit careful about attributing direct uptake of glucose to all those organisms that became labeled.
So, yeah, with the carbon use efficiency experiment where you said basically it doesn't change, it's pretty
Yeah.
constant, and its turnover, can you tell the difference? You proposed death and turnover based on different. Can you tell the difference between, say they assimilate the carbon, and then it's just turnover internally? They're just turning over enzymes and turning over storage compounds without actually dying. So
Yeah, actually, right. It's another place where Paul's modeling has been really powerful because you get specific predictions about, say, production of storage compounds by microbes versus balanced growth in terms of the pattern of isotopomer production of CO2. And we have a paper in soil biology and biochemistry that looked at that and concluded that balanced growth was the best explanation for the patterns we observed. In that case, we only looked at scenarios of storage compound synthesis, and I think some of the other scenarios you're identifying would be fun to play with and ask the model, okay, if you're just doing X, Y, or Z, what should happen to the isotopomers? And his model will provide an answer. I mean, it's actually not a new model that he built. It really is adapted from a long history of pure culture studies that have used this metabolic flux modeling technique, as you probably know. But that would be interesting to think about. Basically, a scenario that makes sense, that's not balanced growth, but some other partitioning of carbon within the cell, and see if the model or the data support that scenario.
or even, let's say you do have balanced growth, like his model predicts, that it's not avoiding the storage organs, but it's simply the turnover of that carbon and replacement that's resulting, that is what consists of that represents your turnover, not death. I'm trying to distinguish between actual cells dying or just the same cell existing in carbon replaced,
Yeah, I don't know. No, yeah, I think if that scenario really is partitioning carbon in exactly the same pattern as balanced growth, I don't see how you could maintain that without the cell getting bigger or dividing, right? What's causing the well, we should talk about that more. Yeah. Go ahead.
I was going to say on that note, that's about all the time we have for questions. I'll let all the further questions for after the talk. But with that, you guys can give Dr. Hungate a round of applause.
Thank you.